3 symptoms. Joint pain that arrived quietly and never fully left, a fatigue that sleep does not fix, and a fog over your thinking that you can almost feel, like you are reaching for thoughts through gauze. If you experience all 3, most doctors have a simple explanation. You're getting older. Each symptom gets its own appointment, its own specialist, its own medication. The research has a different explanation. One source. 3 symptoms. The same cells driving the joint inflammation are releasing chemicals into your bloodstream that reach your muscles and steal their regenerative capacity. The same chemicals are crossing into your brain, where they are pushing your immune cells into a dysfunctional, pro-inflammatory state that disrupts synaptic function and produces the cognitive fog most people quietly accept as inevitable. You already know what these cells are called. In video 4 of this series, we introduced zombie cells. Senescent cells that have stopped functioning but refuse to die, releasing a toxic chemical output called the SASP that damages the tissues around them. Today, we are going deeper. We are going inside the joint, inside the muscle fiber, inside the brain's immune cells. And we are going to show you exactly how one cellular mechanism is running 3 different aging experiences simultaneously, and what the research says you can do about all 3 at once. Most people experience joint pain, fatigue, and brain fog as separate problems because medicine treats them separately. But the research literature is telling a different story. And it has been building that story for a decade. A 2025 review published in the journal Bone Research identified cellular senescence as a central driver of osteoarthritis progression in aging joints. A 2025 Frontiers in Cell and Developmental Biology review from McMaster University named senescent muscle stem cells as a key factor in the decline of muscle regenerative capacity after 60. And a 2024 review in Translational Neurodegeneration identified senescent microglia, the brain's resident immune cells, as a central catalyst in the progression of age-related cognitive dysfunction. 3 separate journals, 3 separate tissue types. One shared mechanism. The SASP chemicals IL-6, IL-1-beta, and TNF-alpha appear in all 3. Not as a coincidence, but because the same zombie cells, deploying the same inflammatory arsenal, are causing damage in all 3 tissues simultaneously. I want to take you inside each tissue, because what is happening there is not what you have been told. Start with the joint. Inside healthy cartilage, chondrocytes, the cells responsible for maintaining the structural integrity of cartilage, are remarkably long-lived. They rarely divide. They exist in a careful, slow-paced balance of breakdown and repair. When chondrocytes become senescent, that balance breaks. Senescent chondrocytes accumulate in aging joints and are present at significantly higher numbers in osteoarthritic cartilage compared with age-matched healthy cartilage. The Journal of Clinical Investigation published a landmark review establishing that senescent cells within joints continue to secrete SASP factors that include not only inflammatory cytokines but also matrix metalloproteinases, enzymes specifically designed to degrade the extracellular matrix, the structural scaffolding that gives cartilage its integrity. In plain terms, zombie chondrocytes do not just inflame the joint. They are actively dissolving the cartilage you are standing on. The University of North Carolina Thurston Arthritis Research Center, in a 2024 review in Osteoarthritis and Cartilage, confirmed that all proposed hallmarks of aging have been observed in articular cartilage, and that age-related changes contribute to the development of osteoarthritis, in part by promoting cellular senescence. And in animal models of osteoarthritis, clearance of senescent cells consistently reduces inflammation and preserves cartilage thickness, with human clinical trials now underway. The connection is not speculative. It is demonstrated by intervention. Now the muscle. The fatigue most people over 60 attribute to aging or to mitochondrial decline, which we covered in video 7, has a third contributor that is less discussed but equally important. Skeletal muscle relies on resident stem cells called muscle satellite cells, or MuSCs, for repair and regeneration. Every time a muscle fiber is stressed or damaged, these satellite cells activate, divide, and rebuild the tissue. This cycle is what allows muscles to maintain mass, strength, and function over a lifetime. In aging muscle, satellite cells become senescent. A 2025 review published in Frontiers in Cell and Developmental Biology from McMaster University found that cellular senescence has emerged as a key factor in the dysfunction of muscle stem cells with aging. Senescent satellite cells lose their ability to activate and regenerate muscle tissue. But crucially, they do not simply become inactive. They secrete SASP factors, including TGF-beta, that impair the regenerative environment around muscle fibers. Think of the basal lamina as the scaffolding around each muscle fiber. TGF-beta thickens that scaffolding until the satellite cells can no longer reach through it to do their repair work, and they push neighboring healthy satellite cells toward senescence as well. The spreading mechanism we introduced in Video 4, zombie cells recruiting healthy cells into senescence, is directly observable in aging skeletal muscle. The result is a muscle tissue that cannot repair itself after stress, that loses mass progressively, that generates fatigue disproportionate to the activity that caused it, and that cannot recover the way it once did overnight. This is not aging in the abstract. This is a specific molecular process operating inside specific cells in your muscle tissue right now. And now, the brain. The brain fog that most people write off as a normal accompaniment to aging has a specific cellular mechanism. And it is not neurons dying. It is the brain's own immune cells malfunctioning. Microglia are the resident immune cells of the brain. In a healthy, functioning brain, they perform essential maintenance work, clearing debris, pruning synaptic connections that are no longer needed, monitoring for threats, and maintaining the anti-inflammatory environment that neurons need to function. When microglia become senescent, they stop doing their job, and they start doing the opposite. A 2024 review published in Translational Neurodegeneration by researchers studying microglial aging found that senescent microglia display reduced process ramification and motility, increased inflammatory cytokine secretion, impaired phagocytosis, and dysfunctional metabolic profiles. They accumulate iron and produce excess reactive oxygen species. The consequence, documented in a 2025 study published in Aging Cell from Nanjing Medical University, is that senescent microglia selectively eliminate excitatory synapses in the hippocampal CA1 region, the area of the brain most directly involved in forming new memories and maintaining cognitive clarity. These cells are not just failing to protect neurons, they are actively removing the connections that enable clear thinking. This is the same hippocampal region the Stanford 2026 Nature study identified as the memory encoding center weakened by aging gut bacteria. Two different mechanisms. One through the gut-vagus hippocampus pathway, one through senescent microglia, reaching the same anatomical target, which is precisely why the brain fog and the memory lapses so often arrive together. And a 2024 review from Frontiers documented a third mechanism. Senescence contributes to brain aging through synaptic dysfunction, neuroinflammation, and impaired neurogenesis, the three processes that, combined, produce what people experience as brain fog, slower processing, and the sense that thinking requires more effort than it used to. This is not vague. This is a specific cellular mechanism with a specific anatomical location producing a specific subjective experience. I want to tell you about a woman I will call Helen. Helen is 71. She came to this series after watching Video 4 about zombie cells. She told us she had spent 3 years managing 3 separate conditions. Her rheumatologist was treating her knee osteoarthritis. Her GP had attributed her fatigue to her thyroid, which was borderline, and she had accepted the brain fog as a consequence of disrupted sleep from the joint pain. 3 specialists, 3 diagnoses, 1 underlying biology. Helen made targeted dietary changes over 4 months. She did not abandon her medical care. She added 4 specific things to her diet consistently. She wrote to us at the 5-month mark. She said, "My knees are not cured, but the inflammation is lower. I sleep better because the pain is quieter. And when I sleep better, the fog lifts. I did not expect one problem to be connected to the other. Nobody had explained that to me." I cannot promise you Helen's experience. Every person is different. These are dietary changes, not medical treatments. Please always speak with your doctor before changing your health routine. What Helen changed, and what the research consistently points to, comes down to 4 things. Food number 1. Strawberries, specifically for their fisetin content. Before the evidence, fisetin from dietary strawberries provides meaningful exposure to the compound, but cannot replicate the concentrated doses used in animal studies. What consistent dietary intake does is contribute to the polyphenol environment that supports the body's own senescent cell clearance. Think of it as a regular input to a system that responds to cumulative signals, not a pharmaceutical intervention. With that context, a 2025 study published in Aging Cell found that fisetin supplementation in aging mice reduced senescent cell burden in skeletal muscle, improved grip strength, and reduced frailty markers, improvements comparable in magnitude to pharmaceutical senolytic agents. In joint tissue, fisetin reduces the production of matrix metalloproteinases from senescent chondrocytes in cell studies, directly targeting the enzymes dissolving cartilage. In brain tissue, fisetin has demonstrated neuroprotective properties in preclinical models, reducing neuroinflammation markers consistent with SASP activity. Fresh or frozen strawberries several times per week. Daily if possible. Food number 2. Red onions and capers for quercetin. Quercetin is the other half of the most studied human senolytic combination, dasatinib plus quercetin, and its tissue-specific evidence is particularly relevant for joint health. A 2024 Phase 2 randomized controlled trial at the Mayo Clinic studying the dasatinib plus quercetin combination in postmenopausal women with osteoporosis produced measurable reductions in senescent cell biomarkers in blood. The same research group has previously demonstrated that clearance of senescent cells reduces joint damage markers in animal models of osteoarthritis. For brain health specifically, quercetin has been shown to inhibit NF-κB activation in microglial cells, reducing the production of the pro-inflammatory cytokines that characterize senescent microglia's SASP output. A 2024 review in Nutrients found quercetin demonstrated neuroprotective effects through antioxidant mechanisms and reduction of neuroinflammation markers consistent with senescent microglial activity. Red onions over white. Capers in salads, eggs, and fish. Apple skin retained. Consistent dietary intake provides meaningful xenomorphic activity, reducing the SASP output of zombie cells even when they are not fully eliminated. Food number 3. Green tea for EGCG. EGCG is the only compound in this series with documented evidence across all 3 tissues we are discussing today. In joint tissue, EGCG inhibits the production of matrix metalloproteinases from chondrocytes and reduces IL-1 beta-driven inflammatory signaling in synovial tissue. A study in rats with induced osteoarthritis found that EGCG supplementation preserved cartilage thickness and reduced senescence markers in joint tissue. In brain tissue, EGCG reduces microglial activation in neuroinflammatory models, lowering the production of IL-6 and TNF-alpha from senescent microglia and supporting the anti-inflammatory phenotype that healthy microglia are supposed to maintain. In muscle tissue, EGCG activates AMPK, supporting mitochondrial biogenesis and improved satellite cell function in preclinical models of muscle aging. 2 cups of plain green tea daily. Decaffeinated retains meaningful EGCG content. Food number 4. Fatty fish. The SASP chemicals driving joint degradation, muscle stem cell dysfunction, and microglial inflammation are all cytokines: IL-6, IL-1 beta, TNF-alpha. Omega-3 fatty acids from fatty fish reduce the production of these cytokines through the resolvin and protectin pathway and through direct NF-κB inhibition. In joint tissue, a 2022 systematic review found omega-3 supplementation associated with reduced joint pain scores and lower synovial inflammatory markers in adults with osteoarthritis. In brain tissue, DHA is a structural component of neuronal membranes with documented anti-neuroinflammatory properties, directly reducing the pro-inflammatory phenotype of activated microglia. In muscle tissue, omega-3s are associated with improved satellite cell activation and reduced muscle protein breakdown in aging adults. 2 to 3 servings of fatty fish weekly. The same recommendation as every previous video because the mechanism it is addressing is the same mechanism. Let me give you the synthesis. This series began by asking a question that medicine has not always asked. What if the symptoms that look like separate aging problems are actually the same biology expressing itself in different tissues? The gut microbiome video showed that forgetting is not a brain event. It is a gut event that reaches the brain through the vagus nerve. The vagus nerve video showed that the communication highway itself matters. The zombie cell video introduced the cells driving damage across all tissues. The inflammaging video named the common fire. The cognitive bacteria video showed that the bacteria you harbor determines whether your hippocampus gets the growth factors it needs. The mitochondria video showed that the energy deficit underneath it all amplifies everything else. And today, for the first time, we went inside 3 specific tissues simultaneously and showed you the same mechanism producing 3 experiences that most people consider unrelated. Your joint pain, your fatigue, your fog, 1 cellular enemy, 3 battlegrounds, 1 dietary response. The series has been building toward this point. There is not a different solution for each problem. There is one underlying biology. It responds to a small number of consistent dietary inputs, and those inputs have been inside this series from the beginning. Here is what I want you to take away from today. The 3 symptoms most likely to define your experience of aging after 60, the joint pain, the fatigue, the fog, are not random. They are not inevitable in their current form. They are not 3 separate misfortunes accumulating. They are one mechanism in 3 locations. And the dietary pattern that addresses that mechanism is the same pattern this series has been building since video 1. Fermented foods, prebiotic fiber, polyphenols, fatty fish, green tea, olive oil, dark berries, quercetin, fisetin. Not as a collection of tips, as a unified approach to a unified biology. So here is your one action for today. Add capers to 1 meal this week. A tablespoon on eggs, a spoonful over a salad. One of the most concentrated dietary quercetin sources available in one of the smallest, most accessible additions you can make. 1 tablespoon, 1 step. The joint, the muscle, the brain. They are all waiting for the same signal. Start sending it. Quick recap. Zombie cells, senescent cells secreting the SASP, are active in all 3 tissues behind the 3 most common symptoms adults over 60 attribute to aging. In joints, senescent chondrocytes secrete matrix metalloproteinases that dissolve cartilage. In muscle, senescent satellite cells lose regenerative capacity and secrete TGF-beta that impairs neighboring stem cells. In the brain, senescent microglia shift from neuroprotective to neurotoxic, selectively eliminating hippocampal synapses. The same hippocampal region the Stanford 2026 study identified as the memory encoding center, weakened by aging gut bacteria. 4 dietary inputs with tissue-specific evidence across all 3. Strawberries for fisetin, red onions and capers for quercetin, green tea for EGCG, the only compound in this series documented across joint, muscle, and brain simultaneously, and fatty fish for omega-3 fatty acids that reduce the SASP cytokines driving inflammation in all 3. Next video? We build the daily protocol. The minimum effective dietary pattern that addresses all 8 mechanisms in this series in a single day's eating. Not a list, a system, with timings, food combinations, and the specific reasons why certain foods work better together than alone. That is the next video. Subscribe button is just below. And if someone in your life has been told that joint pain, fatigue, and brain fog are just part of getting older, please share this series. 1 problem, 3 faces. 1 response. Always speak with your doctor before making changes to your diet or health routine. Everything here is for educational purposes only. Take care of yourself, and I will see you in the next one.